integrated into a pool of latently infected cells. So these cells are mainly memory CD4 T cells. And since the virus production inside of these cells is very low or almost absent, these cells are almost invisible to the other cells of the immune system, like cytotoxic T lymphocytes or natural killer cells. So these cells cannot identify the presence of infected cells and cannot get rid of them. There are multiple potential ways to attack the latent reservoirs. Multiple ways have been suggested so far. The use of therapeutic vaccination, of course, gene editing, chimeric antigen receptor, and many others. So the approach that I'm going to focusing on today involves the use of the so-called latency reversing agents. Now, this approach aims to reduce the size of the latent HIV reservoirs together with the use, of course, of broadly neutralizing antibodies and a therapeutic vaccination. So the idea here is to shock the latent virus inside of the cell so the infected cell becomes visible to the other cells of the immune system so that the other cells, CTLs and NK cells, for instance, can identify it and kill it. So more in details, this is a schematic representation of what I just said. This approach is called shock and kill. Imagine to have a pool of latently infected cells. You would shock them with latency reversing agents, or LRA. Now the virus, which was latent and quiescent before, becomes activated and produced and of course released from the cells outside of in the extracellular environment. Now, since the patient will be still on antiretroviral treatment, the virus could not jump from the infected cells, infecting a neighbor non-infected cells, and the reactivated cells would either die because of viral cytopathic effect, or in a second scenario, which is more likely, I believe, the exposure of the viral antigen on the surface of the reactivated cells will make the cells visible to other cells of the immune system, like CTLs and NK cells, and then these cells would then identify and kill the infected cells. Now, the latency reversing agents that have been studied so far belongs to three major classes: the PKC activators that induce activation of NFATs and NF-kappa B. Those transcription factors are known to induce transcription of HIV-LTR. The HDAC inhibitor, or histone deacetylase inhibitor, and the bromodomain 4 inhibitors. Now, unfortunately, there are some problems related to this classes of agents. So first of all, the PKC activators are known to induce activation of T cells, release of inflammatory cytokines, and so the so-called inflammation process cytokine storm. HDAC inhibitors, and in particular panobinostat, have been shown before to reduce the viability and the cytotoxic effect or functionalities of CTLs and NK cells. And finally, bromodomain 4 inhibitors, even if they're not toxic, they really need to be combined to other agents in order to give some sort of efficiency in the latency reversion. So because of the problems related to this first generation of LRAs, the interest in the shock and kill approach has waned. But just because we didn't find the right LRA doesn't mean that this approach is not worth to be further investigated. So can we try a different approach? Let's take a step back for a minute. So just from the purely experimental point of view, if you have a pool of CD4 T cells that are latently infected, and you want to achieve a strong latency reversing signal, if you want to just experimentally in vitro shock the virus, well, you can do that by simulating the TCR of these cells, and you can clearly use anti-CD3, CD28 antibodies. This would activate the CD4 T cells, and consequently, the latent virus will become activated, produced, and released in high abundance in the supernatant. And this process, as you know, mimics basically T cell activation. So you have 5 minutes. Mimic T cell activation, so the engagement of antigen-presenting cells to naive cells. So this happens when naive cells are activated, becomes and proliferate. And the interesting part is that this process relates to changes in T cell metabolism. Quiescent naive cells moves during this process from oxidative phosphorylation to glycolysis, and these changes are strictly related to change in activity of AKT and mTOR. So can we exploit this approach, these changes in T cell metabolism to reverse latency ex vivo? So we started looking at an enzyme called glycogen synthetase kinase 3, GSK3. This enzyme is always active in quiescent T cells, and as long as it's active, it keeps inhibited a lot of protein in the cells, but also mTOR complex 2. If we inhibit these enzymes with these two molecules, SB216763 and titaglutide, which is already in clinical phase 2 of study, we basically remove the block that GSK3 has on mTOR. mTOR is then free to activate AKT. AKT in turn further activates the downstream signaling of mTOR and also activate NF-kappa B. So glycogen synthetase kinase 3 inhibitors activate AKT and mTOR. Let's give it a try. We did this together with SFGH, the SCOPE team. We took samples from HIV-positive donors on suppressive ART, and for instance, we looked at blood, CD4 T cells extracted from blood, and also cell population isolated from intestinal tissue, gut-associated lymphoid tissue, also from positive donors. So here you see the results of the latency reversion in CD4 T cells from blood. This is the amount of virus released in the supernatant. DMSO is our negative control. Anti-CD3, CD28 is our positive control. And these are the different LRAs that we use. So you see the two GSK3 inhibitors at 1 micromolar induce almost 40-fold more virus than the untreated control. And interestingly, titaglutide, the other GSK3 inhibitors, which is already in clinical phase of study, induce latency reversion also in the guts and gut-associated lymphoid tissue. So these inhibitors reverse latency ex vivo both in blood and in gut-associated lymphoid tissue. But are they toxic? Do they induce T cell activation? So if you treat CD4 T cells for 2 days with these agents or others, you see that there is no change in the cell viability compared to the untreated control, while the HDAC inhibitor panobinostat reduced viability by 20 or 30%. And bryostatin, the PKC activator, has the side effect of inducing activation of T cells by inducing surface expression of CD69, while the GSK3 inhibitors don't. And more interestingly, the GSK3 inhibitors do not induce release of inflammatory cytokines that you can see here, while other LRA, like such a PKC activator bryostatin, do. Now, titaglutide, as I mentioned before, is in clinical phase of study, has already been tested in humans for treatment of neurological disorders, and right now is in phase 2 human trials for treatment of autism and myotonic dystrophy. So we decided to focus our next questions on titaglutide only. And the final point that we wanted to address was, is titaglutide affecting viability and functionalities of CTLs and NK cells? So we have some interesting information coming from the literature. We know that treatment with GSK3 inhibitors of NK cells and CTLs, both in vitro and in vivo in mice models, actually enhance functionalities of these cells. So we decided to look at the same. We did this together in collaboration with the OTT lab. So we took CD8 T cells and NK cells. We treat them for 2 days with titaglutide and other LRAs, and then we artificially activated them with either PMA anumycin or K562 cells that specifically activate NK cells. And then we looked at flow cytometry at activation of the cells, looking at surface expression of CD107A and production of interferon gamma. So let's start with CD8 T cells. Just looking at viability, you see that in the absence of stimulation, the HDAC inhibitor panobinostat strongly reduced viability of CD8 T cells, while titaglutide in the red bar didn't. And if we look at production of interferon gamma, we see a slight increase given by pre-treatment with titaglutide, while bryostatin decreased production of interferon gamma in these cells. And also surface expression of CD107A is slightly increased in the presence of titaglutide. And this is basically what happens also in NK cells. Panobinostat and here also bryostatin are very toxic just for the viability of T cells, and the functionalities are also affected. So concluding, the GSK3 beta inhibitor titaglutide reactivates latent HIV ex vivo in CD4 T cells from donors on suppressive ART and also in gut-associated lymphoid tissue. They do that without activating T cells, and they also preserve the functionalities of CTLs and NK cells. Now, clearly we feel that the jury is still out as long as we don't prove this also in vivo. So what we're doing right now is that we are treating SIV-infected rhesus macaque on suppressive ART together with Afam McCoy and Louis Picker at the University of Portland. And yeah, we hope to confirm this result also in vivo. with this, I like to conclude. There are a lot of people to thank that are listed here, and thank you for your attention.